Mithra this side. Today I am going to present my model Li-Fi speaker. Li-Fi is the light emitting diode which transfers the data to the light receiving panel. Let me show you my model. This is the infrared LED which transmit the data from here to this panel. See, I have this mobile. From this aux cable the data comes here to this LED. It has internal circuits which including a resistance and all the transmitters, and this has the panel. From this the speaker wire goes. Here also we have used 220k resistor, and from there this is the power source we have used that. For that we can heat this like this. We will heat, so all the panel LEDs will shrink. Now we can start our audio. See. See, now it is working. Now I will put my hand in between. See, now it is stopped because the passage of the light is blocked by my hand. When I will remove my hand, see. You can think that it is very slow because of the resistance. I have used 220k because it was available. You can use 520k. From that it will be fast.
Sanitizer dispenser that I made at home. If you also want to make it at home, then the materials required are: first one is the IR sensor or the approximate sensor. Second one is the 12 volt DC to run the sensor. Third one is the 220 volt AC to run the motor. We have also used computer power supply to convert AC into DC.
A disc brake normally contains a push rod or a brake pedal attached to it, a brake pads, and a master cylinder assembly which includes a piston and obviously a disc or a rotor. You can see how the disc is rotating through the vehicle's axle. Now, the men on the other side started pushing the brake pedal. The brake pedal or the piston pushes the disc, and behind that the brake pad stops it. Now, as a person pushes the brake pedal on the other side, which causes a sliding friction against the brake pad and the piston, which in turn stops the moving on.
Now take a glass of water and dissolve 2 to 3 spoons of sugar in it completely. After dissolving it completely, put some yeast in it. Now keep this solution aside for 6 to 8 hours, and after this period of time you can observe that this solution has been converted to ethanol or ethyl alcohol. This conversion takes place due to the anaerobic respiration performed by yeast. Now after 6 to 8 hours we obtain a solution. It contains ethanol, and it contains approximately 70 to 75% of ethanol in it. And after this, take some glycerin and put 2 to 3 spoons of glycerin in it. Here glycerin acts as a moisturizer for our hands. And after this, put few drops of antiseptic liquid in it. It is put into it to get rid of the foul smell. And after that, put some aloe vera gel in it. Aloe vera gel is good for skin. And now thoroughly mix it. Now after mixing it, If foul smell persists, you can add some more antiseptic solution in it. So now our hand sanitizer is
My
ready.
name is Ashnu Singh Kohli from class 9th B. In this summer, to beat the heat, I will teach you how to make a cooler at home by yourself. Material required: a tin can, a battery, and a fan, and a connector, and one motor. We have to take motor and fan and put it on it. And then we require a battery and a connector. We have to put the cap on the battery like this. Join both the connect wires to see that the positive and the negative are apart. We have used the battery, the switch, and the motor and the fan, and we have joined so many wires also. The fan carefully and see it is joining. Now the main step is to connect the battery with the motor. Switch on the button carefully. It's working.
Would like to present my model on excretory system. As we all know that our excretory system consists of a pair of kidneys, a pair of ureters, a urinary bladder, and urethra. So here we'll see the process that how the urine flows. As we can see here, that the urine produced is flown in the kidney and then passes into the urinary bladder with the help of our ureters and then finally comes out of a body through urethra.
Look at my model.
Experiment of isolation of DNA from a banana. For this experiment, you will need a salt. The salt causes the protein and other debris to clump together. This allows the DNA to separate from the other parts of the cell. Now, take some soap that serves as a lysis solution. A lysis solution contains detergent and proteinase K. The detergent disrupts the cell membrane, allowing the proteinase K to break the histones away from the DNA. Now, gently mix the solution. Now add some water into it. Now, grind one banana with 2 or 3 tablespoons of this solution. Gently squeeze the cloth. Now take some amount of this mixture into a test tube. Now pour some chilled isopropyl alcohol in this solution. When we pour the fine solution into the beaker or test tube with isopropyl alcohol, the alcohol clumps the DNA together so it is easy to view with naked eyes. Now let this stay for a while. After a while, you will see a mesh-like structure formed here. That is generally the DNA, which can be visible with naked
Installing
eye.
solar panel in the robot like I have done in my robot. I have take a small motor and connect it with the solar panel by using wires. When the sunlight passes through the solar panel, it converts into electricity and wire conduct the electricity and the robot starts moving.
So I have connected the wires with the solar robot going from the motor. Okay. So now it's time to go through the sunlight and see how solar robot works. You can see how solar robot is going. This is because when the sunlight going towards the solar
About how to identify acid and bases. For this, you would be needing a boiled red cabbage. Here I've taken the extract and some household items like vinegar and baking soda. So you can see here I'm adding vinegar to the extract. As you can see, the color changes to red. This is because it's an acid. Let's see what happened with the baking soda. Here I am taking a dash of baking soda. As you can see, the color changes to green. This is because it's in base. In your case, it might be different because there are some strong acids and some strong bases. Strong acids have brighter red color and strong bases have brighter yellow color.
Global warming. So, in this section, due to global warming, the glacier is melting. And in this section, the plants has cut and the animals are died. In plants, there is no humus and it is not fertile at all. You can see that the pollution is increasing. Due to this, the gases like methane, nitrous oxide, carbon dioxide and more is increasing. Section you can see that the sun is harming the ozone layer of.
So as soon as I would press the switch, the current would flow from the battery to this piece of iron and it would immediately convert into the electromagnet. Now you must know that magnet attracts steel pins towards it. See, it has converted into a magnet. But as soon as I would leave the switch and the electricity would be interrupted, this would again be converted into the iron. So that is how this electromagnet works.
As you can see, I have drawn a dot at the center of the line. Now we'll dip it into the water. Now we can see after dipping for a few seconds into the water, the dyes of different particles of black ink have been separated by this experiment.
Electromagnetism. So let's dive into it. So here I have the model. First of all, let me show you how it works. The coil connected with the battery is the sender coil, and the coil with which the bulb is connected is called the receiver coil. The current flows to the sender coil as it is connected with the battery. The receiver coil is not connected with any source of electric supply, so no current is flowing through it. But when we bring it nearer to the sender coil, the magnetic field lines associated with the coil changes and current is induced in the receiver coil, and hence the bulb glows. Let me show you.